we're pioneering the restoration of neural pathways using our proprietary multi-site direct current stimulation technology that we're applying to ALS and stroke. The problem that we're addressing is that injury to the brain or spinal cord results in increased excitability in motor neurons. And this results in difficulty. It interferes with normal hand and leg movements in the case of spasticity. And in the case of ALS, it reduces motor function, actually leads to motor neuron death. Our solution is a proprietary platform for targeted multi-site neuromodulation designed to restore and preserve movement. We have a veteran team with over 120 years of experience, and we have a world-class board that has joined in the last 2 years. The board includes former president of Medtronic Diabetes, who joined us as chairman. It includes the former chief innovation officer at GE Healthcare. And most recently, we had John Donahue join us, who was the founding director of the Wyss Center for Neuroengineering in Geneva. And his name was mentioned earlier by Jim. So our lead indication that we're focused on is post-stroke spasticity. And as many of you know, spasticity is a state of painful muscle contraction that affects about 30% of stroke patients. And currently, we have very limited treatment options, and these include medications such as baclofen and tizanidine, the intrathecal baclofen pump, there's surgery where you actually cut the nerve or the tendon, and then there's botulinum toxin, which is a potent bacterial neurotoxin and needs to be injected 3 to 4 times a year. So what we've developed is MyoRegulator, which is a new modality for doing non-invasive treatment of muscle spasticity. This is get rid of this. This is a simple point of care therapy that could occur at the bedside, in the clinic, and eventually at the patient's home. And the way it works is that electrodes are placed at multiple points along the neural axis, and it's totally targeted to the muscle groups that we're treating. You have current that is driven across the spinal cord and down the peripheral nerve. And what that does is it suppresses the firing of hyperexcitable spinal motor neurons. So this is definitely not TENS. We're using 1 to 4 milliamp current levels, which are applied for 20-minute treatment sessions. And our trials that we've run have been 5 days of therapy for initial therapy. What this does is it reestablishes neural pathways at the cellular level by normalizing the activity of the spinal motor neurons. Our device, our pre-commercial device, is shown on the right. It's a microprocessor controlled device that uses disposable single-use encoded hydrogel electrodes that we've been developing. We've had a number of publications that we've put out over the years, and we're working to move that forward. Over the last 5 years, we've now been working on 3 different clinical trials. Our US feasibility study was published in 2019. This was in upper limb post-stroke spasticity patients. We saw a statistically significant improvement in both spasticity and motor function with no SAEs. Recently, we've completed our EU pivotal trial, and this was done in France at the Paris Brain Institute. This was a randomized controlled trial done in 44 subjects with lower limb post-stroke spasticity. And this is going to be those 2 trials will be the basis of the CE mark that we are going after in the next 12 to 18 months. We're currently in the midst of our US multi-center pivotal study, and this is in 76 subjects. It's happening at Spalding in Boston, and we are very grateful for support that we have from NINDS. Thank you, Nick, and that's funding that study that we're currently working on. We expect to finish that by 2023. So what we've seen in the European pivotal study is a reduction in spasticity. And this is the primary endpoint, which is a change in ankle spasticity as measured by the modified Tardieu scale. And in the active group, we saw about a 35% reduction in the MTS, whereas in the sham control group, we saw basically no difference. So this was statistically significant and clinically relevant. We also saw improvements in motor function, just like we did in upper limb. We saw it in lower limb, which is very interesting to the neurologists. And we also saw improvement in pain and quality of life. And we're working on a paper that should come out over the next several months. So we're currently working on a financing that is intended for commercial launch of the myoregulator device in Europe and then the US. We're working to get to CE mark clearance in early 2024. This is a class 2A low risk device. We've been, as I mentioned, working with ICM, which is France's leading neurological research institution. The trial was sponsored by INSERM, which is France's equivalent of the NIH. And we've been working with our notified body. Commercial strategy is to launch in France, UK, and Germany initially. The business model is device plus disposable, so where we sell the device plus the disposable single-use electrodes, which ultimately account for the revenue driver. In the US, once we finish the US multi-center pivotal study, we expect to be able to get FDA approval. We expect that to happen in 2024. We do have FDA breakthrough designation on this product, and FDA has also confirmed that the trials we're doing are considered non-significant risk and on the de novo pathway. In the US, as we heard earlier, there's close to 2,000 stroke treatment centers. So we're definitely looking forward to getting this into the hands of the clinicians once it's approved. So far I've been talking about stroke, but where the science has led us relatively recently is to ALS. And ALS, as everyone probably knows, is a rapidly fatal neurodegenerative disease with an average life expectancy of about 3 years. About 100,000 patients in the 6 major countries. There's 6 FDA-approved drugs that, beyond the Amylyx recent approval, they really represent 2 molecules. However, none of these 6 drugs really has much of an impact on either patient survival or on outcome. What's very interesting is that over the last 10 years, it has become clear that motor neuron hyperexcitability is actually an intrinsic part of the ALS disease process. So the motor neurons are overactive, they're firing excessively to the point where the neurons actually die. Then you no longer can move your muscles, you can't breathe, and then the patient dies. So we have a technology that allows us to non-invasively suppress motor neuron overactivity, and we believe this is an entirely new therapeutic strategy for ALS. We've spent the last 3 years generating a robust package of preclinical data, including electrophysiology, biochemistry, motor function, and survival. We've been presenting this at the leading ALS conferences in the world. We're very pleased to have received our first NIH grant. Thank you again to Nick and Emily, who I think is on the call. And that is allowing us to generate additional preclinical data. We also are using that funding to develop an animal use version of our platform for multi-channel stimulation. Just in February, we're very pleased and honored to have been named the recipient of the SURF Medical Prize in ALS for medical electronics. And as we mentioned earlier, this is a UK foundation who is basically looking for technologies that could keep the founder of the foundation alive longer. So just to share with you a couple slides of data, what we've been doing really is to apply the technology to the gold standard animal model of ALS. This is a SOD1 G93A mouse model. We apply electrodes along the spinal column. We have electrodes also on the sciatic nerve and a return abdominal electrode. The mice develop tremor, paralysis, and premature death. Not sure what happened. Remove spotlight. This one here. This one, right? I think it's this one. That's this one, yeah. There we go. Yeah, coming across, great. Yeah, so on the right, you see the mouse positioned in a mouse holder where we could apply stimulation, measure reflexes, and assess other properties of the animal. EMGs can be done, and what this animal model develops by about day 113 is hyperexcitability in the muscles, which you see on the left of figure A. And as soon as you turn on stimulation, you see a very significant decrease in spike amplitude. And after you treat for 3 days, 3 consecutive days, 60 minutes a day, you see that after treatment, and this is when stimulation is turned off, you see lasting decreases in both spike frequency and amplitude. And we've actually looked into molecular mechanisms of what's happening there. So there's one video I wanted to share with you, and this is a study done in stimulated sibling mice. And these are mice that were born in the same litter to the same mother. So these are, siblings. Let me start the video if I can. And what you're seeing is that there are 2 unshaven mice that did not receive stimulation. And then there are 2 mice that have been shaved that did receive stimulation. So it should be apparent that there's a significant difference in motor function between the stimulated and unstimulated animals. We also have data showing increased survival, a whole bunch of data. So at this point, we've been working to refine the designs of the product to enable multi-level spinal stimulation together with bilateral peripheral stimulation. We're looking to get that into a human feasibility study as soon as possible. And we've already designed the clinical trial for this product as well, working with leading ALS specialists. So we're very excited by that. We think we could make a significant impact in the ALS field. So we view the platform as having broad potential. I've so far talked about stroke and I've talked about ALS. However, there are other neurological conditions that have neuronal hyperexcitability as part of the disease. And this includes spinal cord injury, multiple sclerosis, cerebral palsy, brain injury, spastic paraplegia. So we believe this platform is directly applicable to these other conditions as well. We have pioneered the field of multi-site neuromodulation. We have 71 issued patents that actually cover this technology. We have freedom to operate, and we have registered trademarks to the product names. At this point, as I mentioned, we're working to bring together a 25 million Series B for commercial launch in the EU and US. We've been really working the last few years to de-risk the company. We've shown spasticity efficacy in 2 clinical trials. We've confirmed the regulatory pathways in US and Europe. We have manufacturing underway, strong safety profile, and we have actually an FDA green light to start the ALS clinical study. And we've done this all with $2.5 million of investor money raised. The rest of it has been non-dilutive, so far about $10 million. So we're again working to move this forward as fast as we can, and we're very grateful for the support that we've had over the years from numerous organizations that are shown here. Thank you for your time and attention. I'm happy to take some questions.
Thank you very much. This was I'm very excited about the ALS indication, simply because there's really nothing else out there, as you pointed out. We spoke offline a little bit yesterday about the HDE pathway versus traditional PMA. So can you explain for our audience
Yeah.
the route, the regulatory route you want to go with the, with the ALS product, and if you're not doing,
Yeah.
say, how we can accelerate this?
Right. We believe that the pathway for the ALS product will be similar to the pathway that we've been pursuing for the spasticity product. And there, it's not a 510K because there is no predecessor device. But we also do not need to do PMA. So we're doing de novo.
Okay.
We believe it's going to be de novo for the ALS product as well. Again, there's no predecessor
Exactly.
device.
Exactly.
So obviously, we're going to have to be in discussions with the regulatory authorities. We believe they will see the merits of being able to bring a non-pharmacological approach, especially since the ones that exist don't really do anything. So we would like to fast track this and move this as fast as possible. I mean, we've been talking really to some of the world's experts in ALS, and they want this in humans like right away. So that's kind of where we are. Yeah.
And could you explain a little bit more about the hyperexcitability hypothesis?
Yeah.
Is it a hypothesis at this point, and what you would need more to demonstrate this is what the mechanism of action of your therapy would be?
Well, I would say hyperexcitability is more than a hypothesis in ALS. It's actually been documented across various subtypes of ALS. So axonal hyperexcitability is well established. Now, there are periods in the disease where there's relative hypoexcitability as versus hyperexcitability. But what's interesting is our technology actually allows us to modulate in both directions. So by changing polarity, if that was the case, we could actually address it.
It's that simple. Changing polarity
Yeah,
will
yeah.
change you from a hypo
Correct.
to a hyper excitability.
Yes, from anodal to,
intervention.
yes, from anodal to cathodal stimulation along the spinal column. That affects
And
the
so,
motor
yeah, that
neuron
was my
function.
next question: is where would how specific in terms of the localization? Because this is non-invasive, right? This is in terms of where you're placing the electrodes.
Oh, yeah. Well, for spasticity, it depends on which muscle group you're trying to treat. And it's all based on,
for
and
ALS.
for ALS, you're going to want to treat along the entire neural axis. The disease typically starts at one level and propagates
Yes.
in multiple directions. So I think our thinking is, as we get into clinical trials, if patients are having more symptoms, upper limb, I think we'd want to start there. But we also want to be stimulating lower limb as well, just to slow down.
So it would be an
the
evolving
deficits.
therapy where you're starting maybe up, you're starting upper and then moving lower as the disease progresses.
Or vice versa, if they're starting with lower limb dysfunction.
Got it. Very interesting. Do we have any questions from our audience for Nader? Anyone from online? Yes, we have a question.
You mentioned you got a bunch of patents. Question, what name is that under? Is that proprietary? Because I had trouble finding it.
Oh, well, the original technology came, that's okay. Original technology came from CUNY, City University of New York. So that's where the original IP came from our scientific founder, Professor Zagul Ahmed, who's chairman of this department. However, the company has also been developing its own IP.
Thank you.
Yeah.
Okay, anyone? Thank you very much. It was an excellent presentation, very promising. Please keep us apprised of your progress
Thank you.
with both, with both therapies, and we'll be certain to be covering it in our publication.